Hello, guys. First, thank you for the opportunity of being talking here at RECOMB. It's a very great conference, and it's an honor to be here. So I'll talk about a research that I developed during my postdoctoral work at Columbia University. It's about promiscuity in gene expression of horizontally acquired DNA. You guys are going to understand more about this soon, but it's basically about regulatory compatibility among microorganisms. And first, I give a quick overview about horizontal gene transfer. It's a widespread phenomenon among microorganisms, and there is a particularly relevance for clinical case because it's part of the reason for the spread of drug resistance. That's a major problem in public health right now. And one other aspect of horizontal gene transfer that is particular interest is because it changes how we interpret the tree of life. On the left side, you have the traditional tree that is, as it was thought by Darwin over 150 years ago. You have a common ancestor, and all the axon species that we see today, they can be vertically linked to this common ancestor. And when you have horizontal gene transfer, you have a much more convoluted and complex case where the tree of life has branches all over that represents the gene transfer. And one thing that is particularly relevant for this research, we are looking at the regulatory regions. It's not only the gene that has that is transferred, but there is also the regulatory part of it. And there's very little known. The only thing that is known is that they must be incorporated and expressed in a way that benefits the recipient organism. So here's the background for the problem that we are working and how we are approaching it. At the top, I represent a foreign piece of DNA that is carried inside a recipient cell through the process of horizontal gene transfer, that is the HGT there. And this DNA has some coding regions and cis-regulatory elements that is represented by the red and gray, gray colors in the below the figure in the middle. And these cis-regulatory elements must be recognized by the trans-regulatory machinery, which are the sigma factors and transition factors inside the recipient cell. And the expression can have a wide range. If it's low expressed or too high expressed, it may not really provide the fitness that is necessary for the function of the cell. And there is, at some point, an adequate level of expression that would improve the fitness of the recipient organism and would lead to long-term retention of the horizontally transferred elements. And so that was the schematic representation of the problem, and this is how we approach it experimentally. We developed a technique, a method that we call synthetic HGT, where we simulate horizontal gene transfer of regulatory elements in high throughput. So first, we collect regulatory regions that are representative of the diversity of the microorganisms that we take from public database, mostly from NCBI. And this is about 180 different genomes. And we synthesize them in vitro, and we connect to a neutral protein that is GFP, and we transform in high throughput in distinct recipients. There are phylogenetic distinct recipients. The activity level of the regulatory elements is quantified by RNA-seq and DNA-seq, and we chose 3 distinct recipients: Bacillus subtilis, E. coli, and Pseudomonas aeruginosa. They are relevant both in clinical case and also in fundamental biology. They are well studied in general. And they have 2 distinct phylums and distinct GC content. This is just how we define the activity levels, is based on the ratio of the normalized abundance of RNA and abundance of DNA that you observe for each possible promoter element. And we normalize this activity level according to the endogenous to bring something that has more valuable biological meaning. And this is how it behaves when you compare. On the left side, I have phylogenetic tree where I group the promoters according to the donor phylum. And on the right side, I have the 3 possible recipients and the expression level. For this figure, for this talk, one feature that I want to highlight, if you see on the right side, Pseudomonas aeruginosa, you see all the bars are towards the right side and very strong, which indicates a high expression Pseudomonas aeruginosa. E. coli would be something between, and Bacillus subtilis has the least expression of a foreign DNA that comes there. And we want to understand what's driving this different profile, and this is part of the investigation. When you're working with gene regulation, a feature that we look and that we are particularly interested is sequence patterns, regulatory motifs that are in the sequence that could be driven this expression. So we plot the distribution of the transcription activity for each recipient, and we took the 10% most active promoters in all of them, and you look that for all cases, what you observe is the sigma 70 motif, which is the housekeeping sigma 70 in all the three organisms. So a foreign promoter, it comes to the cell and is recognized by the housekeeping, and that's what it's actually driving this differentially expression. And what is relevant, but I'm not going to show a particular figure on this, it's even if you look for alternative combinations to search motif, if you take high express in one, or differentially express among the organisms, or different percentile beans, you still see the sigma 70 or the generator version of the sigma 70 as the signal. So the sigma 70 is the main factor that is recognized a foreign transcript in the cell. And for this talk, I focus on two particularly important findings that we saw, is that this is related with the promiscuity of the transcription. So we call promiscuity how easy is a recipient to express a random promoter that they receive from a foreign DNA. And we see that it's very different in terms of the fraction of promoters that are expressed, and also the multiplicity, how many alternative transcription start sites they can recognize. On the left side, it shows the fraction of promoters that are not transcribed. Bacillus subtilis has a hard time to express a foreign promoter. 71% of the promoters, you see the DNA count there, so it's inside the cell, but you see zero expression. And Pseudomonas is the most promiscuous one, and only 16% of the promoters that you see the DNA there are not expressed. And this is also consistent with the multiplicity of the transcription site sites. It's very common in Pseudomonas aeruginosa to have promoters with multiple transcription site sites. So this is the investigation that we are trying to do and find an explanation, a mechanism that could explain this. And so the model that we're thinking and we hypothesize, you have the sigma 70 motif that is represented in the sequence, and as you can see is 80 rich. And if you have a promoter with a sequence that matches the consensus sequence, you'd expect it to be active. However, you may have case that the sequence is not exactly a match to the consensus sequence, and that would reduce the activity level when you have more degenerated versions of the sequence. And what we're wondering is if this match would be associated with the GC content background of the recipient organism. So this is the schematic model that we're thinking to explain what you observe. We imagine that the recipient machinery, it has to recognize promoters that has a signal compared to the background. You don't want to, it skips a sequence that you could find by chance just by the genome that they have. And then you can compare the sigma 70 motif is a low GC content motif, so it's an 80 rich motif. And you can see two backgrounds. You have the circles, which represent different 80 richness of the promoter, and you'd have the background of the genome that are the blue for the high GC genome and more reddish for the low GC content genome. And as it's clear in this figure, it's much easier to see the promoters and the signal in a genome with high GC content background. So that's what we hypothesized that was happening in the cell, and that's actually what we end up finding. I showed three figures, and the first one is an analytical model where I simulate what would be the chance of finding sigma 70 binding site based on the GC content background of the recipient, and it's based on a multi distribution. And there's two panels on the right side. They represent, I just scan the motif that I find in the genome, and I correct by the background of the sequence. And the y-axis is the fraction of promoters that I would observe, and the x-axis shows if I have a predefined threshold. And in all cases, what you see is that recipients with high GC content, they are much more likely to have a positive signal for diffraction of recipients. So this is only in terms of finding the binding site, and want to see if this also makes sense in the expression level that we observe in our library. And we still see the same sequence. The x-axis is just the threshold that would define what would be active or non-active, and the y-axis would be the fraction of promoters that would be active. And we see Pseudomonas aeruginosa, that is high GC content, would be more likely to express promoter, whatever threshold that you select to consider active or inactive. And just giving what I would call executive summary on the slide, if you want to take one message, a single message from this talk, if there are parts that is confusing, the main message is that the contrast between the GC content of the promoter and the background is what explains the activity level. And this, I'm not going to go into too much detail on this figure because I'm going to run out of time, but there is some interesting model and simulation that we can find that shows that horizontal gene transfer actually constrain the diversity of possibilities of the regulatory code in the microbial world. Because they have to speak the same language, there is an innovation sharing mechanism that they want to, there's a selection towards microorganisms that will explain, that will talk the same language. And then I finished the talk. This work was performed when I was working at Harris Wang Lab at Columbia University. Harris and Nathan was the one that worked direct with me. And now I currently work in Memorial Sloan Kettering. I'm in Van den Brink Lab, so I'm thanking him as well, the lab, to be here. And this work had funding from NIH and NS. And of course, thank you all the audience, and hope you guys enjoy.